Hello, you guys. In this video, we're going to go ahead and transition into the next set of notes for Unit 2, the 2.2 notes. In these notes, we're going to focus in a lot of detail on the cell membrane that surrounds the cell and what the structure of that membrane is and how it's functioning in cells and why it's so important. So we'll go ahead and jump into topic 1 here. In topic 1, we're going to specifically look at the structure of the plasma membrane, first and foremost, structurally what the plasma membrane looks like in a lot of detail. And then we'll kind of do a brief overview of the functioning of the plasma membrane as well. And so here's a nice little picture to start off with of the plasma membrane. This is typical of a plasma membrane we would find in cells. And so you guys will see that the primary component here is this phospholipid bilayer. So you have all these phospholipids with their hydrophobic tails and their hydrophilic heads facing one another, forming this bilayer. That's the primary component of the plasma membrane. But then among that, there's a bunch of other things that you're going to see also in the plasma membrane that we're going to talk about in this video. Just from this picture alone, you can already see like a bunch of proteins embedded in this phospholipid bilayer with cholesterol molecules, which are steroids, and some sugar chains attached to the outside of this membrane. So we're going to talk about each of these different components in a second. But what's important to know right off the bat here is that we do have all these different things embedded in the cell membrane, and all of this is very fluid. So all of these components, like these proteins and these other molecules embedded in the cell membrane, they are all moving freely past one another a lot of the times. And so like this guy right here, he can float on over somewhere else, or these phospholipids are floating among one another and next to each other and moving around. And so for that reason, we call this the fluid mosaic model of the plasma membrane, because there's a lot of fluid motion going on in terms of these components we see here. And so the basic functions of the plasma membrane, which we've kind of talked about a little bit already, is that first and foremost, it separates the internal space of the cell from the external environment. And so this is the barrier that's going to now surround and enclose the cell, and is what's separating it from the outside of the cell or the surrounding environment of that cell. It also controls what moves into and out of the cell, which is super important. In all cells, there's going to be things that need to go into the cell and things that need to go out of the cell. All of that has to take place at the plasma membrane. And then it also allows cells to communicate with one another and to communicate with other cells. A lot of that communication between cells is going to be because of components on their cell membranes. And then, so just jump into these different components here. So like I said, there's the phospholipid bilayer. We've talked about this a lot already. It's just these phospholipids with their hydrophobic tails and their hydrophilic heads forming this bilayer that we see here. So just make sure you guys remember that these phospholipids, they do have a polar and a non-polar region, a region that's hydrophilic and hydrophobic, and they form a bilayer that's specifically orientated in a way where the hydrophobic tails are in the interior of the membrane, and the polar hydrophilic heads are on the outside of the cell and facing inside the cell, which is exactly what we see going on in this picture. And then among those phospholipids and in this bilayer, you're going to have a bunch of proteins. These proteins are everywhere. There's lots of different types of proteins floating around in the cell membrane. You can see a bunch of different ones in this picture right here. Some of those proteins we call peripheral proteins. Peripheral proteins are proteins that are not fully embedded in the membrane. They're usually floating on the interior side of the cell. So we would always find them on the interior of the cell floating around near the cell membrane. And then there's integral proteins. Integral proteins are proteins that are fully embedded within the cell membrane, like this one right here, or this one right here. And a lot of times these are transmembrane proteins, meaning that they go across the whole span of the bilayer from one side to the other side of this phospholipid bilayer. Those integral proteins that I just pointed out, there's a new vocab word here for you guys, which is amphipathic. Amphipathic refers to molecules that have both hydrophobic and hydrophilic regions. And so there's these are molecules that are not completely hydrophobic. They're not completely hydrophilic. There's actually a piece of this molecule that's really hydrophobic and a piece of the molecule that's really hydrophilic. And that's going to be true always of integral proteins. Because they go through the whole membrane, they do have a region that's very hydrophobic that likes to hang out with these hydrophobic tails. But then they also have these regions on the outside and inside of the membrane that are hydrophilic, where the polar heads are and the other hydrophilic stuff is, and where the water is outside and inside the cell. And this is kind of what basically keeps it embedded in the membrane. The fact that this is hydrophobic and wants to stay in here is what's preventing this guy from just popping off, floating somewhere else in the cell. And so just know that that's, know this term from this point on, amphipathic, and that a lot of times when we're looking at these membrane proteins, they are going to have polar and non-polar regions that are allowing them to be embedded in the cell membrane. Another component of the cell membrane is cholesterol. Cholesterol is a steroid. If you guys look closely at this molecule, it has 4 fused carbon rings. 4 fused carbon rings is characteristic of a steroid molecule. Steroids are lipids, and lipids are nonpolar. Cholesterol, like all steroids, is a nonpolar molecule. It's hydrophobic. It doesn't like water, and which makes sense in this picture because look who he's hanging out with. He's hanging out in the inside of this bilayer with the hydrophobic tails away from the water. Cholesterol, by the way, is usually just found exclusively in animal cells. We don't really find these molecules in plant cells. Just a point worth noting. And then their function, like why is there cholesterol? Cholesterol in the cell membrane actually helps to control the fluidity of the cell membrane. So depending on the amount of cholesterol and a bunch of other variables like the temperature that this cell is exposed to, the cell membrane is going to be more or less fluid. So there's some cell membranes that are very fluid and very squishy and flexible, and then there's some cell membranes that are a lot more rigid and not flexible, and that has to do with the amount of cholesterol in those cell membranes. More specifically than that, you don't need to. It gets a little tricky because it depends on if it's colder temperatures or warmer temperatures, if the cholesterol is helping it be more fluid or less fluid. But just know vaguely, right, for this class that the cholesterol is there to help control how fluid the cell membrane is. And so depending on how fluid the cell membrane is, you're going to see more or less cholesterol. That's all you need to know for now. And then another thing you guys see in this picture is these chains attached to some of these proteins and attached to some of these phospholipids. If you look at these blue guys right here attached to the protein, or these green things right here attached to some of the lipids, these are actually carbohydrate chains. These are chains of sugars, chains of monosaccharides, carbohydrates, that these short chains are attached to some of these proteins and some of these lipids. Proteins that have a sugar attached to them, or a sugar chain attached to them, are called glycoproteins. Glyco referring to sugar, and then it's attached to a protein. So we call this guy right here a glycoprotein because he has a sugar attached to him. And then lipids, some of these phospholipids, they have a sugar chain attached to them as well. We call those glycolipids. So glycolipids, glycoproteins, those are just proteins and lipids that have sugar chains attached to them. The sugar chains that are attached to, that we find attached to these cell membranes are help. The purpose for them is to help with cell identity and cell recognition. And so this is what kind of identifies the cells and makes different cells different from one another externally. And so, well, first of all, let me just make sure this is clear. These sugars are always on the outside of the cell. So I would know, just by looking at this picture, I know that this is the exterior side of the cell. This is outside the cell, and this would be inside the cell, because the sugars are always going to be outside the cell. And these sugar chains are going to be different depending on different species, for sure. But then even within the cells of an individual, different cells, like within my cells, different types of cells in my body have different sugars attached to them, or different chains attached to them. And this, I don't know where. Oh, and so it's going to be brought up in a second, but just to say it now, since we're talking about these sugars, they, like I said, they help cells with identifying one another. So when a cell encounters another cell, these sugars, these carbohydrate chains on the outside of the cell, help cells identify one another, so they know what type of cell they're interacting with, or if that cell doesn't belong in that body. So, for example, there's like you guys, I'm sure, are aware that sometimes you can get an organ transplant. Like there's patients who need a new organ, a new heart, a new liver, whatever it is, and you can't just take anyone's liver or anyone's heart or anyone's organ, you have to see if they're a match. And so when you hear that, like on TV and stuff, if they're checking donors to see if they're a good enough match, what they're looking at specifically, one of the things they're looking at is these carbohydrate chains, because if the carbohydrate chains on the cells of this heart that you're receiving as a transplant, if those carbohydrate chains are very different from the carbohydrate chains that are found on your heart cells, then your immune system will actually recognize that those cells don't belong in your body, that these cells are from a different person's body, and your immune system's trained to attack those cells, which usually is a good thing, because when we have bacteria and viruses and stuff like that, we want our immune system to attack those cells. They don't belong in our body. But in the situation where you're trying to give someone an organ so that they can live, we would prefer that their immune system accepts that organ. And so a lot of times we have to be careful and make sure that those carbohydrate chains are a strong enough match in terms of their similarity. And if they're similar enough, they would go ahead and try it and hope for the best and hope the immune system is okay with it and gets tricked into thinking that those are actually the cells that belong in that body. And this has to do also with genetics. So you can imagine that the people who have the most similar carbohydrate chains as you are going to be your immediate family. And so that would be a great place to start looking for a possible match. So that's what those guys are doing. So anyway, here's this magical combination of components that make up this fluid mosaic model of the plasma membrane. And so hopefully you guys understand all these different components and who they are, and make sure you know them. In terms of these proteins that we find embedded on the cell membrane, there's a lot of different types of proteins that are classified as membrane proteins. These are proteins embedded on the cell membrane. I give you 6 different examples in these notes, I think. There's channel proteins and carrier proteins. These things are called transport proteins just in general. They're moving material in and out of the cell. A channel protein has an open channel, hence the name, that specific ions or specific molecules can flow in and out of the cell through. A carrier protein doesn't have an open channel, but instead it actually binds to specific molecules and then through a shape change pushes them to the other side. And so these proteins actually will bind to a specific molecule and then move it to the other side. But both of them are transport proteins moving material in and out of the cell. There's also cell recognition proteins like the glycoproteins that I just pointed out. These are proteins that have carbohydrate chains attached to them, which help cells identify one another. There's receptor proteins. We've talked about these before. These are proteins that are on the cell membrane and they bind to specific signaling molecules outside the cell, and they cause things to happen inside the cell because of those signals that they're receiving. There's also a bunch of enzymes who are embedded on the cell membrane. So enzymes, a lot of enzymes are just floating around in the cell doing chemical reactions or floating around in specific organelles doing specific chemical reactions. And then there's also a lot of enzymes who are actually embedded within membranes, within the cell membrane, where they're going to catalyze specific chemical reactions. And then a last example is junction proteins. These are proteins that help cells adhere to one another. So in the last set of notes in topic 4, we talked about different types of cell junctions like gap junctions and desmosomes and tight junctions. Those are junction proteins in the cell membrane of this cell and the cell membrane of this cell that are holding on to each other. This one looks like a gap junction. There's a channel between them that material can go from this cell to that cell. But all of these are types of proteins embedded in the membrane. And if I asked you on a test, for example, like where in the cell are these proteins made? Like hopefully you know that these proteins were made by ribosomes. Ribosomes put these proteins together. But specifically, these would be made by the ribosomes embedded on the endoplasmic reticulum, the rough ER. That's where these proteins were made because if you remember from last set of notes, the ribosomes on the rough ER are responsible for making membrane proteins. Any proteins embedded on the cell membrane, that's one of the types of proteins that the rough ER makes. So all of these guys that I just talked about, those are all made by the rough ER before they're sent to the Golgi, and then the Golgi sends them to the cell membrane. And then these notes finish with a couple quick ideas here. So this membrane, which is controlling what goes into and out of the cell, we say that this membrane is selectively permeable, meaning that it only certain substances can go in and out or across the cell membrane, in and out of the cell. So some molecules can, some molecules can't, some molecules can more easily go across, some have a more difficult time going across. And so, like I was saying, it's selectively permeable. Certain substances go in and out, and that has to do with the structure of the plasma membrane, giving it that ability to control what substances are going in and out. And so, there's some important big concepts here about what is allowed in and out of the cell. And so, one thing you guys need to know is that for small molecules that are hydrophobic or nonpolar hydrophobic molecules that are small, those molecules can very easily just go across the cell membrane through these phospholipids. So like this guy right here, he's a nonpolar hydrophobic molecule. Molecules like that, they can just actually squeeze their way through the phospholipids to get to the other side. And so those can move freely in and out of the cell if they're nonpolar or hydrophobic. These hydrophobic nonpolar tails in the middle, and they have no problem with these guys trying to move on through to the other side. The problem becomes when we're looking at polar or charged molecules. So charged molecules like ions or polar molecules like water, for example, is very polar. Those things that are hydrophilic are going to have a hard time going through these phospholipids. And so like these ions right here, you guys can see that they're having a really hard time being able to get through these phospholipids. And that's because of this nonpolar hydrophobic center in the phospholipid bilayer. It doesn't allow polar and charged molecules to easily get across. Sometimes they can kind of sneak across. Every now and then one gets through, but for the huge majority of them, they have a really hard time getting in and out of the cell. And so those guys, things that are polar or charged, these hydrophilic molecules, they're going to need the assistance of transport proteins to get in and out, kind of like this picture showing here with water. So transport proteins are usually required to allow these polar or charged molecules to easily go in and out of the cell. And so make sure you know that starting now, that nonpolar things can easily just squeeze their way through in and out through the phospholipid bilayer directly through these phospholipids. But polar or charged molecules, things that are hydrophilic, they're going to have to go through proteins. Proteins are going to have to help them out to get across. And then the last thing on here is talking about cell size and an important concept for this class called surface area to volume ratio. And so cells like to have a lot of surface area. So surface area is the total area of their surface, out like that membrane surrounding the cell, for example, or like in this cube right here, the surface area would be the area of this side and the area of the other surface and the area of this surface, all the surfaces, their total area, that's the surface area. And cells want to have a really high surface area because this is where the membrane, the surface, is where things are able to get in and out of the cell. And so there's a lot of things that need to go in and out of the cell, and that's going to happen way more easily if you have a lot of surface. So if there's a lot of surface everywhere, then a lot of membrane to work with, there's a lot of different places where things can get easily in and out of the cell. That's going to be a more efficient cell if it has a large surface area. But the problem becomes the volume of the cell. So cells actually don't want a huge volume. Volume is how much space is inside the cell. And cells want a small volume because if you have a big volume, if there's a lot of space inside the cell, it's going to take forever for things to move within the cell. We're talking about microscopic molecules, nanoscopic molecules, like on a molecular level, for molecules to go from like one side of the cell to the other side of the cell. If you have a big cell, it's going to take a while. And so that's not as efficient as when you have a small volume. Things can very easily and quickly move within the cell to where they need to go. A good example is like oxygen. You guys are breathing in oxygen right now. It's going to diffuse into lots of cells in your body, all the cells in your body. And that oxygen is going to go across the surface of the cell, the plasma membrane. It's going to move into the cell. And again, if cells have a lot of surface, then there's a lot of places for oxygen to get into the cell, which is good. But once in the cell, that oxygen is going to have to now diffuse, which means like by random movement, get to where it needs to go, which a lot of times is the mitochondria. The mitochondria needs the oxygen to make ATP for the cell. And so if the mitochondria is on this side of the cell and the oxygen is way over here, that oxygen is just moving randomly in the cell, and it's going to take some time for it to randomly make its way to the mitochondria. And if we're talking about a big cell, even though cells are small, once they reach a certain size, it's going to take too long for things to get around. Like through the random movement of all these molecules, once you have a big enough volume, things start becoming way too time consuming. And so cells, they want to have a small volume, so that way things can very easily and quickly diffuse throughout the cell into different parts of the cell. And so with those two concepts in mind, that cells want a large surface area but a small volume, this kind of restricts how big a cell can get. Because if a cell is getting bigger and bigger and bigger, it is gaining more surface area. Like bigger cells are going to have a higher surface area, but it's also going to increase their volume as they get bigger, the space inside. And if you work out the math, which you don't need to work out the math, when cells get bigger, the volume actually increases way more than the surface area. So as cells get bigger, they're increasing way more in volume than they are in surface area, which is less ideal. So overall, like getting bigger actually is a problem because your volume's increasing bigger than your surface area is. And so for that reason, cells like to be small. Like that's why cells are small. Cells are small because small cells have a large surface area for the amount of volume that they have. They have small volumes with more surface area for that given amount of volume. So maybe a better way to explain this is the surface area to volume ratio is what we really care about. Like what's the ratio between the surface area and the volume? We want the highest surface area with the lowest volume, and smaller cells accomplish that goal of having a high surface area with a small volume, a high surface area to volume ratio, way better than bigger cells. And so we're going to do some math in a second, or you're going to do some math in a second. But just memorize this right now, that cells want to have a high surface area to volume ratio. The higher the surface area to volume ratio, the more efficient the cell is at obtaining nutrients and needed material and getting rid of waste from the cell. It's all about having a high surface area to volume ratio. And so if you guys look here, here's 3 examples. Here's like one large cell. Let's just pretend this is one large cell, and then or we could take up this volume and spread it out amongst super small cells, 64 smaller cells. But all 3 of these examples have the same volume, right? This takes up 64 cubic centimeters. This takes up the same amount of space. This takes up the same amount of space. So they all have the same volume, but they have different amounts of surface. So if this is just one cube and you add up all the surfaces, it's going to have a certain surface area. They already did the math for you, so don't worry about it yet. But this is the surface area, and this is the volume. If you do 4 times 4 times 4, this is the volume. And then if you do 4 times 4, which is 16, that's the surface area. That's the area of this surface, and then you would times it by 6 other surfaces. Anyway, you get 96. Now, if we took that cube and divided it up into 8 cubes, what you'll notice is that the volume doesn't change, but now we have way more surfaces. So we have all the surface of this and all the surface of that and all the surface of this, and there's these surfaces in the middle and in between these cells. There's a lot more surfaces. So the surface area goes up a lot. And then if you further divide those cells, you still are taking up the same amount of volume, but by having all of this volume spread out into smaller cells, the surface area went up a lot. And so here I calculate the surface area to volume ratio. To calculate the surface area to volume ratio, you take the surface area and you divide the volume. Let me repeat that. You do the surface area divided by the volume. Please don't do it the wrong way. Don't do the volume divided by the surface area. That's going to give you the volume to surface area ratio, which is not what we want. We want the surface area to volume ratio. And when you take the surface area of this guy and divide it by its volume, you get this number right here, 1.5. Or if you take the surface area of this guy and you divide it by its volume, you get 3. If you take the surface area of this and you divide it by its volume, you get 6. Now what you're going to see is that ratio is going up. And the higher that ratio is, the better the cells are at absorbing and getting rid of absorbing important molecules and nutrients and getting rid of waste products and transporting things in and out of the cell. This is going to be the better situation than this. And so in your notes at the very end, there's lots of questions here for you guys to answer. But at the very end of your notes, there's 3 examples that you guys need to calculate. This is actually on the AP test where you have to be able to calculate the surface area to volume ratio of cells. Usually they give you some pretty easy pretend shapes to work with. And so here's 3 different shapes, this cube versus this rectangular prism versus this sphere. And they want you to calculate the surface area of each of these 3 shapes. And then they want you to calculate the volume of each of these 3 shapes. And then you're going to take that surface area and that volume, and you're going to divide it, surface area divided by volume, to figure out the surface area to volume ratio. And once you know all that information, you will be able to then answer this question. It says, which cell would be the most efficient at absorbing nutrients and excreting waste? And so right here is a snippet from your guys' formula sheet for AP Bio. This is on your AP Bio formula sheet. You guys don't need to memorize these formulas, but you do need to know how to use them. And so if you don't know how to find the volume of this, it tells you to find the volume of a rectangular solid. You just do length times width times height. It should be pretty easy. You can do that for this. You can do that for this to find the volume. To find the volume of this guy, the volume of a sphere is 4/3 times pi times the radius cubed. So figure that out. You've done this since middle school. I believe in you guys. And then for the surface area, to find the surface area of a sphere, this is the formula. Figure out what that's going to be. To find the surface area of a cube, you can just do one of these sides is 3 times 3, so that would be 9. That's the area of this side, and then you just times it by 6, or just follow what this formula is saying. Now, the hardest one is actually the rectangular solid, to find the surface area of this, because there's different sized surfaces. There's this surface in the front, which is 2 by 4. This is 2 by 4 micrometers. So to find the area of that, you just do 2 times 4. So I'm doing this for you almost. That's 8. And then the backside is the same dimensions as this. So the backside would also be 8, the surface area. And then it looks like the top and the bottom are also the same. This is also 2 by 4 up here, which is 8 square micrometers. And then the bottom that you can't see is also 8. And so there's 4 sides that have a surface area of 8. So you would add that all up. But then the left and the right side of this picture is a square, actually. It's 2 by 2. That's 4. And then this other side is also 2 by 2, which is 4. So anyway, add up, find the area of each side, and then add it all together. That's your surface area. But anyway, go ahead and calculate the surface area to volume. Well, the surface area of each of these, calculate the volume of each of them, and then figure out their surface area to volume ratio and see which one is the highest. When you figure out the ratio, please write it as a number. Some students leave it as a ratio, but actually write it as a number, like to a decimal place. That allows you to way more easily compare which one's the lowest or the highest surface area. Anyway, that's it. Thank you guys. See you later.